Untangling the mysteries of cellular reproduction, all cells come from cells. Welcome, curious minds, to today's intriguing exploration into the world of cellular reproduction. In this captivating lecture, we will uncover the marvels of cell division and to emphasize the resounding principle, all cells come from cells. This is Cell and Molecular Biology lecture series, and this has been Sir Steven, and welcome to this All Cells Come From Cells presentation, or cellular reproduction, or otherwise known as cell division. Cell division, the cornerstone of life, perpetuates the continuity of living organisms. At its core lies a fundamental truth that every cell originates from pre-existing cells. This principle, championed by Rudolf Virchow, has paved the way for our understanding of life's perpetuity. From the elegant dance of mitosis, ensuring an accurate duplication and distribution of genetic material, to the dynamic narrative of meiosis, heralding genetic diversity, and these processes showcase the symphony of life encoded within every cell. The cell cycle is the process by which new cells arise from other living cells, which is we call the cell division. That certain tissues are existing or certain tissues have continuity throughout the life takes place with very differently in prokaryotes and eukaryotes. There are two types in eukaryotic cells. We have mitosis, which leads to the production of cells that are genetically identical to their parent and as a basis for producing new cells, or we call it the somatic or the autosomes. For meiosis, it leads to the production of cells with half the genetic content of the parent, which is reductionist approach based for producing new sexually reproducing organisms, or we call it the gametes or the gametic, which are the sex chromosomes. These are the phases of your cell cycle. The cell cycle is the series of stages that a cell goes through. In the M phase or mitotic phase, it includes the process of mitosis and cytokinesis. Mitosis lasts about an hour or so, where cells divide. In an interphase, the G1, the synthesis phase, and the G2 phase, which majority of the cell cycle will take place. It lasts longer than the M phase. It will happen days, weeks, or longer depending on the genus or the species of the organism. It has a diverse metabolic activities. The gap 1 takes place between the end of the mitosis and the beginning of DNA replication. DNA replication occurs during the S or synthesis phase. The G2 occurs between the end of S and the beginning of mitosis. S phase length can be calculated directly from the percentage of the cells whose nuclei are radioactively labeled during a brief pulse with thymidine. And you will see here the hours after the thymidine addition. It increases after 5 hours, and it peaks at somewhat 8 or 9 to 10 to 12 hours after the thymidine has been added, and it goes down as we progress to the 20th hour, and it goes back again when it reaches 25 and above. Now we talk about the cell cycles in vivo. 3 cell types are distinguished based on their capacity to grow and divide. Cells such as nerve cells, muscle cells, or the RBCs that are highly specialized lack the ability to divide. Cells that normally do not divide but can be induced to begin DNA synthesis and divide when given an appropriate stimulus, such as your lymphocytes or your hepatocytes or liver cells. Cells that normally possess a relatively high level of mitotic activity, stem cells for blood elements such as skin and other epithelia, even plant apical meristems. Stem cells have an asymmetric cell division in which the daughter cells have different fates. Now, to control your cell cycle, it's focused on the initiation of DNA replication and the initiation of mitosis. A replicating cell contains factors that stimulate the initiation of DNA synthesis. A cell will undergo mitosis, will contain factors that trigger, again, chromosomal condensation. We have identified this one using cell fusion experiments where G2 to M transition under positive control. So you will see here the G1 chromosomes, your mitotic chromosomes, and some of them are pulverized chromosome fragments, and the transitions between the G1 to S, S to G2, and G2 before M. And the control of cell cycle are regulated by what we call as your protein kinases. When the cells are about to enter the M phase, they are triggered with the activation of a protein kinase promoting factor, or the MPF. They have two subunits, a kinase and a regulatory subunit, which is a cyclin. When cyclin has been increased, it will activate the kinase. You will see that in the graph where there is an MPF activity and the cyclin activity high and low. The protein kinases have a very important role in the cell cycle. MPF-like or cyclin-dependent kinases, CDKs, are studied in yeast TS mutants. The products of your CDC2 or fission yeast and CDC28 genes are CDKs, which are responsible for passing through G1, the start, and G2, which are the control points, where the start is the cells are committed to DNA replication, which is in the middle of the G1 and the S cyclins, and the G2 to M, where cells are going to enter the process of mitosis, where there are mitotic cyclins. With cyclin binding, the levels of particular cyclins rise over time, and different cyclins are being transcribed at the different points in the cell cycle. When this cyclin is reaching a sufficient concentration in the cell, it binds to the catalytic subunit of the CDK, which will cause a major change in the conformation of the enzyme's active site. Cyclin binding will cause the movement of a flexible loop of CDK polypeptide chain away from the opening of the active site, will allow the CDK to phosphorylate its protein substrates. With CDK phosphorylation or dephosphorylation, it will happen in late G2, where the mitotic cyclin CDK becomes activated and mitosis is triggered. You will see a yeast cell on your right, where two kinases and a phosphatase are very important in the change of the CDK activity. It's like a lock and key analogy. With CDC activating kinases will phosphorylate both a threonine and tyrosine on a CDK subunit. A phosphatase removes one phosphate, and the singly phosphorylated cyclin is activated during the cell, driving the cell to mitosis. So this is your figure, which shows you a progression through the fission yeast cell cycle that are requiring the phosphorylation and dephosphorylation of critical CDC42 residues. So this in interphase in G2, and then in the second phase where the CDC25 has been activated, and then the CDC2 kinase and cyclin is being activated. It removes a tyrosine, and then in G1, the post-mitotic fission yeast cells will become inactive as cyclin has been degraded. Cells with a mutant WEE1 gene cannot maintain the CDK in an inactive state and divide at an early stage in the cell cycle, producing smaller cells, hence the name WEE. Cells with mutant CDC25 gene cannot remove the inhibitory phosphate from the CDK and cannot enter mitosis. The balance between the WEE1 kinase and CDC25 phosphatase activities is being regulated by still other kinases and phosphatases. This is your progression of, again, the fission yeast cell cycle, which requires the phosphorylation and dephosphorylation of critical CDC42 residues. With CDK inhibitors, it stops the formation or attachment. In budding yeast, the SYK1 protein acts as an CDK inhibitor during the G1. The degradation of SYK1 initiates DNA replication. With controlled proteolysis, what we call as ubiquitin proteasome pathway, where there are two classes of multi-unit subcomplexes, which is the SCF and the APC function as ubiquitin ligases. We destroy the mitotic cyclins and allowing the cell to exist mitosis and enter a new cell cycle. CCF ubiquitin ligase will enable the entry into the S phase by promoting the degradation of the S phase cyclin-dependent kinase inhibitor SYK1. With subcellular localization, you will see it here, a dynamic phenomena in which cell cycle regulators are being moved into different compartments at different stages. So the major mitotic cyclin B1 shuttles between the nucleus in the cytoplasm until G2 when it accumulates in the nucleus. Then the pairing between individual cyclins and CDKs is very specific and only some combinations are found. In yeast, CDK1 is the only CDK required to drive a mammalian cell through the cell cycle. They have also important proteins in mice such as CDK1, SICB1, or SICA1. Loss of it is lethal and others are being compensated for by the others. So these are the combinations of your cyclins and CDKs as they progress through in the cell cycle. Mice that are unable to synthesize CDK1, cyclin B1, cyclins E1 and E2, or cyclin A2 will die as early embryos. A mouse embryo that lacks the genes encoding all the other cell cycle CDKs, such as the 2, 4, and 6, will develop to a stage with fully formed organs, although the animal does not survive to birth. You will see this the roles of cyclin CDKs in the mammalian cell cycle, showing you a mouse embryo. As we progress through the cell cycle, they can be arrested at a checkpoint. How they can be arrested? There are three factors. The first is the sensors that detect chromosomal abnormalities. One good example of that is your aneuploidy. Okay, the unjust distribution of chromosomes for every cell. We have also transmitters that signal the information and the effectors that inhibit the cell cycle machinery. CHK inhibits CDC25 when DNA is damaged at the G2 to M transition. So really important to note that before the cells to exit the interface, they must be checked with CHK1 that will inhibit the CDC25 when the DNA has not been properly and faithfully replicated. The protein P21 will inhibit the G1 CDK when DNA is damaged at the G1 to S transition. So we see here some of the models for the mechanism of action of the two DNA damage checkpoints. So it can be affected with ultraviolet radiation or ionizing radiation. So these are the control of the cell cycle. We have checkpoints, CDK inhibitors, and cellular responses. Progression through the S phase is inhibited by the action of P27, which is what we call as a CDK inhibitor. The P27 forms a trimeric complex with Cyc A and CDK2, preventing their activity. Elimination of this cell cycle progression inhibitor causes excess cell proliferation and organ growth in P27 knockout mice. So this is your P27 again, a CDK inhibitor that arrests the cell cycle progression. With the Anaphase, we have mitosis and cytokinesis. Mitosis is a process of nuclear division in which two nuclei with identical genetic content to the parental cell are produced to maintain a chromosome number. We have mitotic stages: the prophase, prometaphase, metaphase, anaphase, telophase. Each phase is being characterized by a particular series of events. Each of these stages are representing a segment of a continuous process. So for prophase, chromosomal material condenses to form compact mitotic chromosomes. These chromosomes are seen to be composed of two chromatids. They are being attached together at the centromere. Cytoskeleton is disassembled and mitotic spindle is assembled. Golgi complex and endoplasmic reticulum fragments where the nuclear envelope disperses. For the prometaphase, chromosomal microtubules attached to the kinetochores of the chromosomes. Chromosomes are moved to the spindle equator. For the metaphase, chromosomes are aligned along the metaphase plate attached by chromosomal microtubules to both poles. For anaphase, centromeres will split and chromatids will separate. Chromosomes move to the opposite side poles. Spindle poles will move further apart. And lastly, in telophase, chromosomes cluster at the opposite spindle poles. Chromosomes become dispersed, where a nuclear envelope then will assemble around. It will enclose all the chromosome clusters. The Golgi complex and the ER will reform, and daughter cells are being formed by cytokinesis. For a very detailed explanation, prophase will be the formation of the mitotic chromosome. The duplicated chromosomes are prepared for segregation. The machinery for mitosis is being assembled. The chromosome compaction and condensation occurs in early prophase. Condensin is a protein which is responsible for compaction. Where compact chromosomes appear as a rod-like structures, you will see that on your right. Each mitotic chromosomes will consist of two chromatids. Prior to replication, each of the DNA chromosome is being associated with cohesin, which forms a ring to encircle the two sister DNA molecules or the chromatids. Cohesin and condensin will both contain a structural maintenance of chromosomes, proteins. These proteins fold back on themselves to form a highly elongated antiparallel coiled coil with a globular domain at both of the N and the C termini. So this is the model for the role of condensin and cohesin in the formation of your mitotic chromosomes. For cohesin, this is a this is very distinct and has two unique phases or stages, with most removed during the prophase. We have polo-like kinase and aurora B kinase phosphorylate cohesin, and the protein WAPL then removes it. Chromatids are held relatively loosely along their extended arms but much more tightly at their centromeres. So each of the mitotic chromosome is comprised or compromised of a pair of sister chromatids connected to one another by the protein complex cohesin. Cohesive. Centromeres occur at the primary constriction on chromosomes and serve as the binding site for proteins. Kinetochores are on the outer surface, which is found on the outer surface of your centromeres. They are the sites where the chromosomes attach to the microtubules of the mitotic spindle. So you will see here a centrosome shown by the arrow here next to your nucleus. And this is the outer kinetochore. It has a microtubule binding, a microtubule motor activity, and processes the signal transduction. In your inner kinetochore, the centromere will replicate, the chromatin has interface, and the kinetochore forms. And we have here the centromeric heterochromatin, centromeric heterochromatins, the kinetochores, the microtubules, inner plate, interzone, outer plate, fibrous corona, and a lot of proteins, dynein, corona fiber, CNPE, the depolymerase microtubules, NDCAT in outer kinetochore plate. So this is the plus end and the minus end. The plus end, again, remember, of the microtubules is where the subunits are added or lost. If you are checking that one out, please check the video Cytoskeleton and Cell Motility. When they enter mitosis, the microtubules undergo a disassembly before reassembly into the mitotic spindle. Disassembly is from inactivation of proteins that stabilize microtubules, such as your MAPs, and the activation of proteins that stabilize them. The centrosome cycle progresses in concert with the cell cycle. So this is a centrosome cycle of an animal cell. In early G1 phase, in S phase, and the mitosis. Separase become activated late in mitosis and cleaves a protein link holding the centrioles together. At the beginning of mitosis, the centrosome splits into two adjacent centrosomes, each containing a pair of a mother-daughter centrioles. This is the centrosome cycle of an animal cell in fluorescent. During prophase, the centrosomes move apart from one another as they organize the bundles of microtubules that form the mitotic spindle. In the mitotic spindles of the animal cells, your microtubules are arranged in an aster, or it looks like a flower, around each centrosome. So here on the top is the formation of your mitotic spindle. Below is the formation of a spindle pole in the absence of centrosomes. So in general, the prophase is the dissolution of the nuclear envelope and the partitioning of cytoplasmic organelles. Major components of the nuclear envelope are disassembled in separate processes via phosphorylation of key substrates by cyclin B CDK1. Nuclear pore complexes are disassembled, nucleoporine subcomplexes are disrupted, and the subcomplexes dissociate. Nuclear lamina is disassembled and the depolymerization of laminal filaments. Nuclear membranes are disrupted. Mechanically, their holes are torn into the nuclear envelope by cytoplasmic dynein molecules. Membranous organelles such as mitochondria, lysosomes, and peroxisomes remain relatively intact through mitosis. The Golgi complex may have become incorporated into the ER during the prophase, or they may become fragmented to form a distinct population of small vesicles that are partitioned between daughter cells. Now, mitotic spindle is formed. The chromosomes are moved by microtubules into the center of the cell. So we call it the metaphase, the met equator. A kinetochore will contact stabilizes microtubules. A single kinetochore is being attached to microtubules to form the spindle, both of the spindle poles. So this is syntelic attachment. There's this mono-oriented chromosome. There's no tension. Syntelic attachment, no tension again. For bi-oriented chromosome, they have under tension. And lastly, tension is applied by microtubules on the opposite poles of the cell. For prometaphase, chromosomes arrive at the center of the spindle by differential growth of microtubules at the opposite poles. Forces for movements are generated by promoter proteins associated with kinetochores and the chromosome arms. So in this figure, the consequence of a missing motor protein on chromosome alignment during prometaphase, and showing you in fluorescence a microtubule behavior during the formation of your metaphase plate. So you will see that the light green stuff in this micrograph is your chromosomes, and the spindle fibers are in red. Chromosomes are moved by a process called congression. Shortening and elongation occur primarily by loss or gain of subunits at the plus end of the microtubule. The plus end of each microtubule remains attached to the kinetochore. Microtubules from each pole are equivalent in length. So there's an engagement of chromosome during prometaphase and its movement to the metaphase plate. During metaphase, chromosomes are aligned at the spindle equator on the metaphase plate. We have also three microtubule groups. First is the astral microtubules. They radiate from centrosomes to regions outside the spindle body. We have chromosomal microtubules. They move chromosomes to the poles. We have polar microtubules that maintain the integrity of the spindle. So this is the mitotic spindle of an animal cell. In metaphase, again, the microtubule flux in the metaphase spindle involves the tubulin that are treadmilling towards the poles. Subunits are incorporated at the kinetochores of the chromosomal microtubules and the equatorial ends of the polar microtubules. They are lost from the minus ends of the microtubules in the region of the poles. Next is your anaphase, the role of proteolysis in progression through mitosis. They have two distinct complexes, the SEF and the APCs, that will add ubiquitin to proteins at the different stages of the cell cycle. SEF acts during interphase. Anaphase promoting complex, or what we call as your APC, regulates events during mitosis depending on the adapter protein present. So this is the SEF and APC activities during the cell cycle, and they are inversely proportional to each other. You notice in G1, in S, in G2, and M, they increase or they decrease their quantity. Destruction of securin releases an active protease, separase, which cleaves the SCC1 subunit of the cohesin molecule that holds the sister chromatids together. So they have to split. APC-CDH1 completes the ubiquitin of your cyclin B, which causes a drop in activity of the mitotic CDK with the cyclin B CDK1 and the progression from mitosis and into the G1 phase of the next cell cycle. So this is your SEF and APC activities during the cell cycle. The importance of protein degradation in regulating the events of mitosis and the re-entry of cells into the G1 is best revealed with the use of inhibitors. No destruction of cyclin B or proteasome inhibition, the cells will remain in the late stage of mitosis. Subsequent CDK1 kinase inhibition will allow the return to normal activities. The mitosis and cytokinesis will continue. This is an experimental demonstration of the importance of proteolysis in a cell's irreversible exit from mitosis. And this is just a time-lapse presentation of the cells that are being pulled apart together. The chromosomes will form this cleavage furrow until the two cells are being formed from a single cell. Chromosomes are split in synchrony. Anaphase 1 will move the chromosomes towards the poles. Anaphase B, the two spindle poles move in opposite direction due to the elongation of microtubules. So this is what the figure will show you: the mitotic spindle, the greens, and the chromosomes at anaphase in deep purple. Anaphase will continue in two different phases, and the chromosomes are being split in synchrony. Dynein and kinesin are found in the kinetochores of chromosomes. Depolymerization of microtubules will generate a sufficient force to move all the chromosomes. In yeast cells, a protein is pushed by the force released from depolymerization to help move the chromosome toward the spindle pole. So this is an experimental demonstration that the microtubule depolymerization can move attached chromosomes in vitro in 0, 51, and 75 seconds. Anaphase will require forces for chromosome movements, and the KMN network forms at the outer kinetochore of each chromosome. A protein complex known as NDC80 forms a weak linkage with an attachment microtubule just behind its plus end. Terminal heads of the NDC80 complexes are able to travel along the microtubule towards its minus end. They are being pushed by the curling protofilaments of the disassembling tip. This is a proposed mechanism for the movement of chromosomes during anaphase in animal cells. So we have the spindle poles, the depolymerases, the microtubules in blue, the NDC80 complex in orange, the depolymerase in, again, the green, and the outer plate of your kinetochore being pulled toward the plus end. In anaphase spindle assembly checkpoint, it operates at metaphase or anaphase transition to check for misaligned chromosomes. Unattached kinetochores contain a protein complex that sends a weight signal to prevent an entry into the anaphase. If a cell cannot postpone the chromosome segregation, the daughter cells may receive an abnormal number of chromosomes or aneuploidy. So this is the spindle assembly checkpoint. Lastly, in M phase, the telophase is the final stage where the daughter cells return to interphase. The mitotic spindle has disassembled, and the nuclear envelope of the two nuclei are reassembled. Chromosomes will become dispersed. This is your nuclear envelope reforming, and cleavage furrow that initiates or depicts the moment of cytokinesis. We have also motor proteins which are required for mitotic movement, such as they have been powered by what we call as your motors, like dynein and kinesin related proteins. They are located at the spindle poles and the kinetochores. This motor protein has to keep the poles apart, being chromosomes, to the metaphase plate and keep them there, elongate the spindle during the anaphase B. So these are the steps of proposed activity of motor proteins during mitosis. Cytokinesis, again, is the terminal end of your M phase, where the process of one cell being divided into two daughter cells. The contractile ring theory will suggest that a thin band of actin and myosin filaments will generate the force to cleave the cell. The site of attachment, assembly, or the plane of cytokinesis is being determined by a signal coming from the spindle poles. Bipolar myosin filaments composed of myosin 2 are essential for the ring contraction. Forced generating mechanism is similar to actin-myosin mechanism during the contraction of muscle. Sliding of the filaments of the contractile ring will pull the cortex and attach plasma membrane towards the center of the cell. So we'll see here different experimental demonstration of the importance of myosin in cytokinesis. The furrow forming or furrow initiating proteins. The cleavage furrow position is determined by the anaphase mitotic spindle, leading to the activation of row A in a narrow ring within the cortex. The signal for plane determination is thought to travel from the spindle poles to the cell cortex along the astral microtubules. So this is the site of formation of the cleavage plane and the time at which the cleavage occurs depending on the position of your mitotic spindle. So it comes with different forms, shapes, and sizes. With engineering linkage, what are the roles of the membrane tension in cell division? One way cells can sense that they need to divide is through the mechanosensitive ion channels. The piezo channel deforms the membrane to form a curved dome shape. When the membrane is being pulled, the membrane tension is produced and the dome flattens. This is opening ion channels. The calcium ion entry activates cyclin B G2 to M transition. This is your piezo 1, which this is not a globe. It trimerizes into a membrane binding dome shaped channel. Next is the meiosis. During meiosis, the chromosome number is halved and 4 daughter haploid cells are formed. 2 cell divisions will happen without the intervention of DNA replication. Meiosis 1, where homologous chromosomes pair and then segregate, ensuring that the daughter cells receive a full haploid set of chromosomes. In meiosis 2, sister chromatids are separated. This is a visual representation of the stages of meiosis, showing you on your right. Meiosis can occur at different points in the life cycles of eukaryotic organisms. For example, gametic meiosis, the processes being linked to gamete formation such as for animals and protists. For zygotic meiosis, the process occurs after fertilization, such as your protists and your fungi. For sporic meiosis, the process is independent of gamete formation and fertilization, such as your plants and algae. So this is a comparison of the three major groups of organisms based on the stage within the life cycle at which meiosis occurs and the duration of the haploid phase. For gametic meiosis in male, spermatogonia that are committed to undergo meiosis become the primary spermatocytes. Then two divisions of meiosis produces four relatively undifferentiated spermatids. Each spermatid undergoes a complex differentiation to become the highly specialized sperm cell, or what we call as your spermatozoon. So this is the stages of your gametogenesis in vertebrates, formation of your sperm. In females, oogonia becomes primary oocytes, which then enter a greatly extended meiotic prophase. Vertebrate eggs are typically fertilized at a stage before the completion of meiosis, usually at the metaphase 2. Meiosis is completed after fertilization, while the sperm resides in the egg of the cytoplasm. So these are the stages of your gametogenesis in vertebrates where the eggs are being formed. Meiosis has stages. Prophase 1. Just like mitosis, they have prophase, but it's very specified into 1 and 2. Zygotine, where is the second stage of prophase 1. Homologous chromosomes pair. The process is called the synapsis. Homologs associate via the synaptonemal complex, which is a ladder-like structure with transverse protein filaments connecting the two lateral elements. Synapse chromosomes form a bivalent or tetrad, this is your synaptonemal complex. Packetine 3 synapsis will end. Diplotin 4, which is the synaptonemal complex that disappears, and homologous chromosomes will start moving apart. The chiasmata are the remaining points of attachment between two intervening homologous chromosomes. Chiasmata occurs when crossing over took place. So these are the visible evidences of crossing over. So this portion here, they have purple or lavender portions, and they have the orange portions. They come from the maternal and the paternal sides. With the stages of meiosis to continue, Cytokinesis 5, chromosomes are prepared for attachment to the spindle fibers, ends with the disappearance of the nucleolus and the disassembly of the nuclear envelope. This has been triggered by an increase of your maturating promoting factor activity. This is the visible evidence of your crossing over. The two homologous chromosomes are aligned in the metaphase plate. Homologous chromosomes are held by one or several chiasmata. Absence of chiasma can lead to the abnormal segregation of chromosomes. So this is separation of homologous chromosomes during meiosis 1 and separation of chromatids during meiosis 2. In Anaphase 1, there is a stage where the homologous chromosomes will separate. Maternal and paternal chromosomes of each tetrad segregate into two daughter cells independent of other chromosomes. So this is the separation of the chromosomes, homologous chromosomes during meiosis 1 and separation of chromosomes during meiosis 2. With this, the telophase 1 produces less dramatic changes than the telophase of mitosis. The nuclear envelope may or may not reform during this stage. The stage between the two divisions is called the interkinesis between meiosis 1 and 2. Cells during this stage have a haploid number of chromosomes and a diploid amount of DNA. Meiosis 2 is very simple than meiosis 1. It generates cells haploid in both amount of DNA and chromosome number. During metaphase 2, chromosomes are aligned so that kinetochores of sister chromatids face your opposite pole. Sister chromatids separate during anaphase 2. With genetic recombination, it increases genetic variability, mixes maternal and paternal alleles. It happens by the physical breakage of and the ligation of individual DNA molecules, as what you are seeing here, a proposed mechanism for genetic recombination initiated by double-strand breaks. Prior to recombination, DNA homologs are aligned by homology search. Breaks are introduced into one strand of each duplex at the corresponding sides and widened. Then the two duplexes are joined to each other by holiday junctions. These are pair of DNA crossovers. As we conclude this glimpse into the mesmerizing world of cell reproduction, remember that every life story begins with a cell. Join us in the future lectures as we continue our voyage into the intricate realms of cell and molecular biology, with delving deeper into the mysteries that govern the life itself. Thank you so much for embarking on this enlightening journey. Until we meet again, keep exploring, keep questioning, and remember, all cells come from cells. Thank you so much for watching.